Thank you very much. Thanks for the invitation. And although we can't see it outside, the solar eclipse is currently reaching its maximum. And I think that the seminar, instead of darkening our minds, has actually enlightened us a lot, and I hope to contribute to this too. So we are interested in flexible goal-directed behavior, and it has proven very fruitful to investigate errors because they can trigger a broad range of adaptive mechanisms. But the general question is, in fact, how does the brain know when to adapt its behavior, when adaptation is needed? And adaptation can be seen as part of a feedback loop. So when we do some actions, during actions and after the actions, the performance monitoring system is assumed to monitor for any events that indicate that the outcomes of the actions and the states of the organism and the environment are deviating from the goals. And whenever this is the case, this information is used to signal the necessity and magnitude and perhaps also the type of adaptation to other brain regions, which then implement the adaptation. Let me first recapitulate, although I think most of you know them, the correlates of performance monitoring. And very consistently, it has been found that this monitoring and signaling function is associated with activity increases in the posterior mesial frontal cortex that encompasses the anterior mid-cingulate cortex, sometimes also called dorsal ACC, the pre-supplementary motor area, and adjacent dorsal-medial prefrontal cortex. In the event-related potentials, we can see at different stages or different instances of performance monitoring quite similar sequences of negativities and positivities. First, there is a frontocentral negativity followed by a frontocentral positivity, and later on there is a more parietal sustained positivity. For example, for response errors, we can observe the error-related negativity and reaction time tasks. So the ERN reaches its maximum about 50 to 100 milliseconds after the erroneous response and is followed by a frontocentral error positivity and later on a parietal error positivity. And it's suggested to be generated in the posterior mesial frontal cortex. So the error-related negativity seems to reflect single pieces of objective evidence speaking for a performance problem, whereas the error positivity has been suggested to actually reflect the accumulation of subjective evidence and is associated with conscious perception of the error. In other tasks where we have a lot of uncertainty, we need external feedback to disambiguate whether the outcome is correct or incorrect. And then we can observe the feedback-related negativity, FRN, which is then followed by a frontocentral P3A and a later P3B. And during actions, when sudden problems occur, difficulty is higher than expected, and we have to invest effort, then we can see an increase in the stimulus-locked N2 component, again followed by a P3A and P3B component. So what signals are actually reflected in these measures that we can record non-invasively? So let me come back to this loop and unpack this a little bit. So the loop starts with actually and ends with the state of the environment and the state of the organism. And according to our goals and homeostatic needs, we want to achieve certain states, and we have to find out when they actually differ from our needs, when there are changes that actually are threatening our goals. So in order to detect this, we need to make predictions, and we have heard about this a lot. So these predictions can be either model-free, based on simple reinforcement and association learning rules, or they can be model-based, based on prior beliefs and forward modeling. And the actual signal that we can derive from this is actually comparing the state variables and the predictions, and we find a number of prediction errors. So for example, there's the unsigned prediction error, which reflects the magnitude of the deviation and is related to surprise and can indicate the importance of the event. Then there are lots of signed prediction errors indicating the direction of the deviation, whether we are faster or slower than expected, whether we are closer or further away, and so on. And this can help us maybe to choose the corrective action to compensate our performance problem. And a special case is a signed reward prediction error, which indicates the valence of the outcome, whether it's better or worse than expected. So in order to derive an adaptation signal from these prediction errors, we have to weight them appropriately. And I will talk later about different factors that contribute to this weighting, because it's not every single event is maybe important enough to drive big adjustments. So sometimes the environment is so noisy that we cannot always say whether a prediction error signal is really important and should trigger an adjustment. So we can distinguish several kinds of adjustments. On the one hand, there are general, usually task-independent adjustments that are related to the classical orienting reflex. So there are changes in arousal and alertness, autonomic responses that prepare the body for more complex reactions. There are changes in affect and expressions of affect, like cursing and frowning when people make an error. And there's motor inhibition that is related to the classical post-error slowing effect. And these general adaptations may prepare the organism for more specific adaptations. And there is a broad range of specific adaptations that depend on the context, on the task at hand, and at our beliefs on the reasons for the problem and how we can influence the problem. And most studied has been the changes in selective attention and learning from errors, which then leads to updates in our value representations, in our models, and later on in action selection and decision making, and also, again, acts on the predictions. And finally, of course, these adaptations are aimed to change the state of the organism and the environment according to our needs. Well, I will talk in the rest of the talk about the prediction error representations in the brain, how we weight it, and if there's time enough, I will also talk about a study on the signal from the performance monitoring system to perceptual areas to increase selective attention. So since the discovery of the error-related negativity in 1990, this area of research has become really a hot topic, and lots of theories and models have been suggested. They all converge on the notion that the posterior medial frontal cortex indicates the need for adjustments, but they differ slightly with respect to the signals that are represented in the posterior medial frontal cortex and how they get there. I won't go through all these details, and to those of you who are not directly involved in performance monitoring research, they may also appear quite similar. One thing that is interesting, perhaps, is that they predict that we would find certain signals in the performance monitoring system, like signed and unsigned prediction errors, response conflict, outcomes, and weighting factors. Let's first talk about response conflict, because this is a theory that has dominated the debate in the first decade of this century massively, has been debated a lot. And it suggests that actually response conflict is the competition between two or more concurrently activated response tendencies. It's measured by the Hopfield energy, which is a multiplicative measure of the activations in the response channels. And predictions from this model are actually in line with many, but not all, behavioral, EEG, and fMRI findings in simple interference tasks. However, there are some problems with this model. It cannot explain activity in a performance monitoring system when there is no motor response, like with delayed feedback. And I'm not aware of any single cell recording study that has found activity that is related to performance monitoring signals in the performance monitoring, conflict monitoring signals in the strict sense. There were several high-ranking publications, the most recent in Neuron a month ago by the Platt Lab. But I think these studies have not found activity that is predicted by this model. They have used the word conflict, but it's not response conflict. So it could be that what we actually see in EEG and fMRI at the systemic level is a result of the co-activation of different neuronal ensembles just coding for the different action possibilities. And it remains to be seen whether this signal is actually an epiphenomenon or can be used somehow for control. And as we have heard yesterday in Thomas Goschke's talk, there are models that actually don't need this monitoring aspect here and still can explain post-conflict adaptations. Well, what about the reward prediction error? For more than 10 years, people have provided provided evidence for and against the notion that the error-related negativity and the feedback-related negativity reflect reward prediction errors. And we have used multiple point-wise multiple regression approach in a probabilistic learning task. This is feedback-logged activity. And we see activity in the EEG that is correlated with the signed reward prediction error, particularly here in this time range of the FRN. There's a central positive effect that means unfavorable outcomes. Negative reward prediction errors are associated with negative going EEG, so this is reflecting the feedback-related negativity. But one has to look whether this is just a correlation with the outcome or it also correlates with a prediction. So we decomposed the prediction error signal into outcome and expected value, and as you can see, and one would expect for a prediction error signal, both are coded in the opposite directions. So in addition, Telmi and colleagues have used an axiomatic approach adopted from economics and shown that the feedback-related negativity really reflects a reward prediction error. Just on a side note, we also gave fictive feedbacks when people learned what they could have got had they chosen a different option. And interestingly, there is no feedback-related negativity in that condition. We can discuss this maybe later. At the systemic level, we see prediction error signals most likely stemming from the anterior mid-cingulate cortex, but we also find in other studies that unsigned prediction errors elicit activity in the anterior mid-cingulate cortex. For example, when we provided subjects with unexpected valence-free outcomes, they got an N2 activity and also in the fMRI activity in the anterior mid-cingulate cortex. And this has been also shown at a single neuron level. For example, Matsumoto and colleagues have found neurons in the cingulate sulcus of monkeys that coded positive reward prediction errors, negative reward prediction errors, or unsigned prediction errors. And recent work seems to suggest that neurons in the cingulate cortex are actually able to code many different kinds of these prediction error signals, and they're multiplexing this according to the context needed by the current task. So where do these signals come from? Interestingly, in the dopaminergic midbrain neurons, also prediction error signals have been found, like mostly by Wolfram Schulz, but many other people as well. And usually people talk about the reward prediction errors, positive signed prediction errors that are coded in these, by these neurons. But again, more recent work suggests that at least a subset of these neurons also codes unsigned reward prediction errors that are there for positive as well as negative outcomes. So we have, again, both signals here. And the original version of Clay Holroyd's reinforcement learning model of the ERN suggested that actually this prediction error signal is conveyed to the anterior mid-cingulate cortex, where on a negative prediction error, dopamine would be released less, and that would disinhibit pyramidal cells and then lead to the ERN. However, this is extremely unlikely because the dopamine action is way too slow and way too sustained to explain such sharp responses as we see in the EEG. So there are certain alternatives that one can discuss. There may be co-release of glutamate from the same neurons. There may be GABAergic fibers projecting from the VTA to the AMCC, which are fast enough to drive this information transfer. But there may be also top-down control that actually the prediction error signal is sent down from the cortical regions to the midbrain. And finally, as we find prediction errors actually in many places in the brain, it could be that these signals evolve more or less independently. And so far, I'm not aware of any invasive study having simultaneously recorded from the VTA and the cingulate neurons, such that we cannot say anything about the directionality of information transfer so far. Well, what about the weighting? We have to weight this prediction error signal. And in classical Rescorla-Wagner type reinforcement learning models, this is done via the learning rate alpha, which weights the prediction error. So this is the update term. And we modeled our data in this probabilistic reinforcement learning task using a changing learning rate that decayed as learning progressed. And this is the correlation of the EEG activity with the learning rate. And you see that there is a sustained positive centroparietal learning rate effect that's maximal actually at the latencies between the FRN and the P3A. So the more, the higher the learning rate, the more positive is going as the EEG. But again, we don't know where this exactly comes from. So there is evidence that the cingulate cortex actually may be a point of convergence of these weighting signals. So a study by Steve Kennelly and colleagues has shown that lesions in monkeys in the cingulate sulcus actually impair the use of reinforcement history. So these monkeys could only use outcome information from the most recent trials, but not from previous trials. So they could not weight the current outcomes by previous experience. Also, fMRI studies have used different ways of varying uncertainty and have found that actually the anterior mid-cingulate cortex codes learning rates. And these factors that influence the learning rates have been discussed recently in lots of papers. And there's one nice paper by McGuire and colleagues, recent paper, that has actually looked at different effects. So on the one hand, we have expected uncertainty or volatility, which is related to surprise and, of course, the stochastic nature of the outcomes. It cannot be reduced. And it helps us when we have a representation of this uncertainty, because any event that deviates from this normal distribution is highly surprising and may indicate a change point and increases the change point probability, so that something has changed in the environment and we might have to adjust our internal models. On the other hand, we have reducible estimation or relative uncertainty, or it's the opposite of the confidence signal we have discussed yesterday. And this can be reduced with learning over time. It's the uncertainty about our internal models. But in addition to this, even rewards that had nothing to do with the task itself, they carried no information whatsoever, were driving up the learning rate. So there's the question whether any other forms of salience can actually influence our learning. And so McGuire and colleagues have shown that these different factors influencing learning rate are actually represented in different brain areas. Just look here, we have talked about this yesterday. And they converge on a network that comprises the anterior mid-cingulate cortex, the anterior insular and parietal cortex, and the functional connectivities with these regions varied according to the context and to the state of the task. So if I have some time left, I would now switch gears and talk about actually the adaptation signal that's being sent out from performance monitoring to influence selective attention at a trial-by-trial basis. So the idea of this adaptation is that we have to focus on the task at hand. And on the one hand, we have to enhance important inputs that are useful for the task performance, and we have to suppress any other distracting inputs. So in a previous study, we have found that error-related activity in the posterior medial frontal cortex predicted signal increase, post-error signal increase in brain areas that were task relevant in that particular task, and at the same time suppressed activity in areas that were coding distractor information and helped us to adapt behavior. But one question that remains open is, of course, how is this adaptation signal transmitted from the posterior medial frontal cortex to the perceptual cortices? Animal research suggests that acetylcholine plays a major role in attentional regulation. And indeed, there are many projections from the posterior medial frontal cortex to the basal forebrain in humans, the nucleus basalis of Meinert, which is the main source of cholinergic fibers to the cortices. And studies in rats have shown that stimulation of cingulate neurons projecting to the basal forebrain or the basal forebrain neurons in themselves have facilitatory effects in visual cortex areas, and these effects can be blocked with muscarinic antagonists like atropine. And a more recent optogenetic study has actually provided further evidence for this. And anatomical work suggests that the basal forebrain is organized in a modular fashion, allowing task-specific modulation that are needed for selective attention. So we set out to test this in humans by means of a double-blind, placebo-controlled pharmacoFMRI study. So 30 male participants received either saline as a placebo or the muscarinic antagonist biperidone intravenously, and then went to the scanner and did the task. And we had an 8 days washout time to ensure that there was no carryover effects. So we based our hypotheses mostly on the interaction between the posterior medial frontal cortex and the visual areas, because we did not expect to find good signals from the basal forebrain. Cells are very sparse there, and indeed the signal is very noisy that we can find in the fMRI there. So for the placebo condition, we would of course assume to replicate our previous study to find intact post-error adjustments, enhancement in task-relevant visual areas, suppression in task-irrelevant visual areas, and this should also somehow be related to behavioral adaptation. If we block muscarinic receptors, we would hope to find reduced or even abolished post-error adjustments of attention. So this is the task we used. It's a modified version of the Simon task. Relevant stimulus feature was the color, and location in always outward direction of the arrow were irrelevant features that had to be ignored. So this is compatible. This would be an incompatible trial where we expect higher reaction times and higher error likelihood. In addition, people also did a separate color localizer task. Let's look at the behavioral data. There were no treatment effects on error rates and Simon interference effects in this task. Biperidone generally slowed reactions down by about 30 milliseconds. More interestingly, biperidone treatment abolished trial-by-trial adjustments, post-error slowing is gone, and more interestingly, post-error reduction of interference is gone. So in the placebo condition, after errors, the interference of Simon interference effect was reduced significantly, but not with biperidone. We used a combination of independent component analysis and deconvolution in order to be able to disentangle the sequence of error trials and post-error trials and the overlapping hemodynamic responses. So what you see here are three independent components overlapping with the posterior medial frontal cortex, and they show error-related activity. So in this graph, you see amplitudes of the activity in these independent components, trial by trial, from the 5th pre-error trial to the error and afterwards 5 trials. And you see there's an isolated signal increase on error trials in the placebo and biparidine condition, suggesting that this region is really monitoring errors. Then we found components that were overlapping with activity in the color localizer task. This is the color localizer activity, and here are the overlapping color processing independent components. So they should code the task-relevant information. So with placebo, we found on the error trial some reduction of the activity, which is in line with the idea that people have actually focused their attention not so much on the task-relevant information. Then it took about 2 or more trials to actually regain full activity. It's a little bit surprising how slow this is, but it's very similar to our previous study. So, well, we have no clear explanation for that. But interestingly, the error signal in the performance monitoring system correlated with this slope of this increase, post-error increase in activity in color-sensitive cortical areas. With biparidine, this reduced activity is actually regaining full speed or full power much slower and much less intensively. I mean, we now look for the correlation. There seems to be no interaction between posterior medial frontal cortex error-related activity and this increase in activity in color-sensitive areas. Behaviorally, we could predict post-error reduction of interference effects from this slope of this increase in post-error activity in visual areas only in the placebo condition, not in the biparidine condition. We have no good means to really say what visual areas are task irrelevant, but we defined it very loosely by looking at all independent components that were not overlapping with the color localizer. This is an example. There were 6 of these independent components. This is a collapsed time course. You see a gradual slight increase. It's not significant, suggesting that actually people were paying more and more attention to the wrong stimulus features, and then there's a steep decrease on the post-error trial. And again, there was a correlation of error-related activity in the posterior medial frontal cortex with this decrease, post-error decrease in visual areas. And that was also present in the biparidine condition, but it was weaker than with placebo. So to summarize that, we could replicate that error-related posterior mesial frontal cortex signals predict post-error attentional regulation, that there's enhancement of task-relevant visual information processing and suppression of distracting information processing, and blocking central muscarinic receptors impairs behavioral and neuronal post-error adjustments. So this strongly suggests that acetylcholine mediates these trial-by-trial post-error adjustments in selective attention, which may also in part explain the contribute to the beneficial effects that we see with treatment of choline esterase inhibitors, or also low-frequency deep brain stimulation and nucleus basalis of Meinert in Alzheimer's patients. So let's conclude. We have talked about the performance monitoring signals. So it seems that performance monitoring in a network that is associated or is centered around this posterior medial frontal cortex is involved in detection and evaluation and weighting of expectancy violations, and transforms this information into some adaptation signals. What I did not talk about today was the costs of control and effort that is also influencing activity in the posterior medial frontal cortex. And also, there are aversive events that can be predictable. Pain, for example. They deviate from our goals, but they not necessarily always produce a prediction error in the strict sense, but still activate this region. And then this is transformed into some adaptations of our actions, but also of our beliefs, predictions, and also of our metacognition about the uncertainties on this that we have, and this is strongly probably related to error awareness. And it's still quite an open question what the role of error awareness is in post-error adjustments. So with this, I come to the end. I would like to mention the people from my labs in Magdeburg and Nijmegen and the collaborators, and I would like to thank you for your attention.